Endogenous mitochondrial NADPH fluorescence can predict lifespan by Christopher S. Morrow, Palis Yao, Carlos A. Vergani Jr., Praju Vikas Anical, Paula Montero Lopez, Jeffrey W. Miller, Berenice A. Benayoun, and William B. Mayer. In Communications Biology, Abstract, many aging clocks have recently been developed to predict health outcomes and deconvolve heterogeneity in aging. However, existing clocks are limited by technical constraints, such as low spatial resolution, long processing time, sample destruction, and a bias towards specific aging phenotypes. Therefore, here we present a non-destructive, label-free and subcellular resolution approach for quantifying aging through optically resolving age-dependent changes to the biophysical properties of NADPH in mitochondria through fluorescence lifetime imaging (FLIM) of endogenous NADPH fluorescence. We uncover age-dependent changes to mitochondrial NADPH across tissues in C. elegans that are associated with a decline in physiological function, and construct non-destructive, label-free and cellular resolution models for prediction of age, which we refer to as mito-NADPH age clocks. Mito-NADPH age clocks can resolve heterogeneity in the rate of aging across individuals and predict remaining lifespan. Moreover, we spatiotemporally resolve age-dependent changes to mitochondria across and within tissues, revealing multiple modes of asynchrony in aging, and show that longevity is associated with a ubiquitous attenuation of these changes. Our data present a high-resolution view of mitochondrial NADPH across aging, providing insights that broaden our understanding of how mitochondria change during aging and approaches which expand the toolkit to quantify aging. Introduction. As individuals can vary remarkably in their rate of aging, generating effective strategies to quantify aging has become a priority of neuroscience research. In response to this need, many groups have generated aging clocks by using machine learning to create models that provide a prediction of age based on biomarkers of aging. For example, aging clocks trained on DNA methylation, blood plasma protein, or transcriptomic profiles exhibit the capacity to predict chronologic age, the time an organism has been alive, with relatively high accuracy 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. While these techniques represent powerful approaches to quantify aging, these approaches also remain limited by a variety of technical constraints that can restrict their use. For example, approaches involving next-generation sequencing can take months to complete. Further, many existing techniques also involve destruction of the sample, limiting spatial resolution of the resulting data and the ability to track the same organism longitudinally. Many existing approaches also involve intricate sample processing steps that could confound results. Lastly, although many existing techniques resolve a variety of features of aging, each clock individually captures a narrow subset of aging biology, e.g. epigenetic changes during aging. Thus, it is important to expand the diversity of approaches to gain a comprehensive view of aging. To address these challenges, we explored the capacity of nicotinamide adenine dinucleotide phosphate, NADPH, representing spectrally indistinguishable NADPH and NADH, FLIM to track mitochondria across age throughout an intact organism. Using C. elegans as a model of aging, FLIM of metabolic cofactors has been used across many model systems both in vitro and in vivo as a label-free, non-destructive and subcellular resolution technique for monitoring the biophysical properties of NADPH in living samples 12, 13, 14, 15, 16, 17, 18. NADPH FLIM capitalizes on the observation that as autofluorescent metabolic cofactors, such as NADPH, are used differentially by cells, their optical properties will change. For example, NADPH is autofluorescent when reduced, but not when oxidized as NADP+19. Further, NADPH has a longer lifetime when it is bound to a protein, as opposed to when NADPH is freely diffusing in the cytosol 20. Pairing NADPH FLIM with C. elegans is particularly powerful, as C. elegans are translucent and progress through an entire life cycle in 2 to 3 weeks, allowing for rapid imaging of NADPH autofluorescence across the entire body throughout aging. Despite the wide use of NADPH FLIM as a tool across many model systems, only a small number of studies have briefly explored NADPH FLIM in C. elegans, with focus largely restricted to C. elegans development 21, 22, 23, 24. Thus it remains largely unclear how NADPH film could be used to study adult C. elegans, and whether this technology could be used to track aging. Here, we expand upon these initial investigations to establish NADPH film in C. elegans as a non-destructive, label-free and subcellular resolution tool for organism-wide tracking of mitochondrial NADPH, and apply this technology to present a high-resolution landscape of mitochondria throughout aging, which we use to spatiotemporally deconvolve age-dependent changes to mitochondria, and develop approaches to non-invasively quantify aging at the cellular level. Results: we first set out to determine whether NADPH autofluorescence could be used to visualize mitochondria in C. elegans. As expected, throughout tissues in C. elegans, we found autofluorescent signals reminiscent of mitochondria in our NADPH channel, which we conservatively call the blue autofluorescence, BA, NADPH channel, to account for NADPH autofluorescence as well as autofluorescence detected in this channel which may not be NADPH X, 750 nanometers 2p, M, 400 to 480 nanometers, Fig, S1AD. To determine whether these signals were mitochondria, we examined BA NADPH autofluorescence in transgenic C. elegans harboring proteins localizing to the outer membrane of mitochondria, fused to green fluorescent protein, GFP, TOM 20A1 to 49, GFP and TOM 70, GFP. We found that enriched autofluorescent signals in the BA NADPH channel exclusively represented mitochondria in all tissues except for the intestine, where mitochondrial BA NADPH signals were detected alongside autofluorescent gut granules, which were previously shown to be lysosome-related organelles, LROs, Fig, S1AD 25, 26, 27, 28. Moreover, we found that BA NADPH autofluorescence alone was sufficient to visualize mitochondrial lumens in all major tissue classes in C. elegans except for neurons, where mitochondrial matrices are much smaller and lack the space for NADPH autofluorescence to accumulate for visualization consistently, Fig. S1AD, verifying that these BA NADPH mitochondrial signals were predominantly NADPH. We found that a short treatment with carbonyl cyanide p-trifluoromethoxyphenylhydrazone, FCCP, which drives oxidation of NADPH and thus a decline in NADPH Fig. S1E, resulted in a strong reduction in BA NADPH autofluorescence inside the mitochondria of body wall muscle, BWM, cells, and that this effect was largely dependent on mitochondrial respiratory chain complex I, Fig. 1AB29. Further, we observed similar emission of BA NADPH autofluorescence in C. elegans mitochondria and pure NADH fluorescence in solution, Fig. S1F. Finally, we found that C. elegans with a deletion of T20D 3.5 SLC25A51, the predicted C. elegans homologue of the mitochondrial NAD plus transporter MCART1 SLC25A513033132, had strongly reduced mitochondrial BA NADPH fluorescence, Fig. S1GH. Thus, NADPH autofluorescence can be used as a label-free marker to visualize mitochondrial lumens across tissues in C. elegans. To increase the resolution by which we could track NADPH in mitochondria, we next used a FLIM-capable 2-photon microscope to perform FLIM and examined fluorescence signal decay in mitochondria in the germline, the metacorpus of the pharyngeal muscle, PM, BWM, and hypodermis of C. elegans, Fig. S2AG. Mitochondria are dynamic and thus, in many cell types, it is often not possible to track individual mitochondria by FLIM, as mitochondria may move out of an analyzed region of interest in the approximately 1 minute of time required to acquire an BA NADPH FLIM image. However, in C. elegans, we observed that mitochondria were relatively less dynamic, with most mitochondria not moving out of a specified region of interest during data acquisition, Fig. S2C. Thus, we determined that C. elegans mitochondria could be analyzed on an individual level. To quantify the lifetime data, following convention in the field, we used weighted two-component and exponential reconvolution modeling to account for the freely diffusing and enzyme-bound NADPH lifetime components, respectively 16 Fig. S2A. This analysis quantitatively extracts 4 coefficients that define NADPH lifetimes and hypothetically model the biophysical parameters of NADPH. The alpha 1 coefficient represents the proportion of diffusing NADPH. The The alpha-2 coefficient represents the proportion of bound NADPH contributing to the observed lifetimes. Tau1 reports the lifetime of diffusing NADPH, and Tau2 reports the lifetime of bound NADPH. These values can be compressed into one value referred to as the fluorescence lifetime, Tau-M16. Thus, this imaging and analysis strategy allows for non-destructive, label-free quantitative tracking of mitochondrial NADPH across individual mitochondria in live C. elegans. With a validated system for performing BA NADPH FLIM in C. elegans, we next asked whether we could track mitochondria across aging using BA NADPH FLIM. We found that C. elegans harbored detectable mitochondrial BA NADPH autofluorescence signals which were sensitive to FCCP treatment that can be observed in many tissues up until day 11 of adulthood, a time in the C. elegans life cycle that is well after the onset of many phenotypes of aging, such as a decline in locomotion, fertility, food consumption, and resilience to stress, and just prior to when many C. elegans in a population begin to die, Fig. S3ae. Therefore, to track mitochondria across age, we imaged wild-type WT BWM cells in C. elegans at days 1, 3, 5, 7, 9, and 11 with NADPH FLIM. Fig. 1c, supplementary data 1 and 2. Excitingly, we identified many changes in mitochondrial BA NADPH FLIM endpoints in BWM cells throughout the course of aging, Figs. 1 e.g. S3FH. For example, we found that BA NADPH alpha-1 decreased during aging, whereas BA NADPH Tau2 increased during aging. Interestingly, BA NADPH FLIM endpoints remained relatively stable throughout young adulthood and began to progressively change starting around day 5 to 7 of adulthood, a time when C. elegans are known to progressively sustain a general collapse in many physiologic functions. This finding suggests that BA NADPH lifetimes may be linked to organismal fitness, and that some components of mitochondria may not begin to change during aging until middle age, similar to the dynamics of other age-dependent changes in C. elegans 33. Confirming this association between shifts in BA NADPH lifetimes and a decline in physiologic function, we found that BA NADPH lifetimes began to change as C. elegans began to sustain a reduction in egg production, motility, and pharyngeal pumping rate, Fig. 1h. We also noted that as C. elegans became older, individuals became more variable, with some individuals displaying more youthful BA NADPH lifetimes while other animals displayed more aged BA NADPH lifetime profiles, Fig. 1i. Thus, BA NADPH FLIM can be used to non-invasively track mitochondria during aging, and BA NADPH lifetimes may be a proxy for organismal fitness. As we uncovered an age-dependent shift in BA NADPH lifetimes that progressively developed throughout aging, we next used least absolute shrinkage and selection operator, lasso, regression, to construct a model for prediction of age based on BA NADPH FLIM endpoints. As expected, our model provided increases in predicted age as chronologic age increased, Fig. 1j. Matching our data, we observed that our model was unable to discriminate between C. elegans at young ages, as BA NADPH lifetimes did not start to change until later in aging. However, starting at day 5, our model predicted progressively higher ages on average at each subsequent chronologic age time point. We also found that our models predicted more variance in age at later time points, suggesting that these models may be resolving heterogeneity in the rate of aging. Therefore, we next hypothesized that this spread in BA NADPH FLIM endpoints reflected heterogeneity in the rate of aging, and that BA NADPH FLIM endpoints could be used to predict fitness. Many studies have previously demonstrated the capacity to predict remaining lifespan in transgenic animals based on fluorescent reporter expression patterns in midlife, such as lipid droplet number 34, RNA splicing patterns 35, heat shock reporter expression 36, microRNA expression 37, and red autofluorescence of whole C. elegans 27. In addition to these systems, similar to a previous report that predicted remaining lifespan based on motility in midlife 38, we found that separating C. elegans by mobility at day 7 was sufficient to predict remaining lifespan, with higher mobility, C. elegans living longer than lower mobility, LM. C. elegans, Fig. 2ab. To determine if natural variance in motility is associated with changes to BA NADPH FLIM endpoints, we performed BA NADPH FLIM, on the BWM of cohorts of HUM and LM-C elegans at day 7, and found that many BA NADPH FLIM endpoints were significantly different between these two groups Fig 2CD, almost completely reflecting changes we saw in BA NADPH FLIM endpoints in young and old C. elegans, with the exception of BA NADPH alpha 1, which showed a consistent but non-significant trend. Importantly, we observed that HUM C. elegans clustered closer to young C. elegans, while LM C. elegans clustered closer to old C. elegans, Fig 2e. To determine whether BA NADPH FLIM could be used to predict whether C. elegans were either in the HM or LM group and thus whether they would be relatively shorter or longer lived, we trained random forest classifier models with day 7 HUM and SLBA NADPH endpoints, and found that our model provided higher performance in predicting remaining lifespan, area under the receiver operator characteristic ROC curve, AUC, 0.78, Fig. 2F39. Predicting age with our BWM regression model, Fig 1J, revealed that LM-C. elegans had a higher average predicted age compared to HIM C. elegans, Fig 2G. Finally, to determine whether BA NADPH FLIM could be used to directly predict remaining lifespan, we imaged BWM cells of day 7 adult C. elegans individually using BA NADPH FLIM, and then recovered animals and tracked lifespan, Figs 2HJ S3IJ. We observed that C. elegans with the highest BWM cell BA NADPH fluorescence lifetimes were associated with the shortest remaining lifespan, validating the direct predictive power of BA NADPH FLIM to determine future lifespan. Thus, BA NADPH FLIM represents a label-free and non-destructive approach to track aging at cellular resolution and can determine C. elegans motility and predict future lifespan. We next asked whether we could analogously use BA NADPH FLIM to track aging across other tissues in C. elegans. To this end, we imaged and analyzed BA NADPH lifetime endpoints across PM, hypodermal, germline, and BWM cells during aging, and used these data to construct lasso models for prediction of age across cell types in C. elegans, Fig. 3ae. Interestingly, we found that across age, all tissues analyzed sustained largely similar changes to BA NADPH FLIM endpoints with relatively strong correlations in BA NADPH lifetimes across tissues within the same animal throughout aging, and similar to our BWM cell data, we found that the changes we resolved did not begin until middle age, Fig 3CD. Thus, on a global level, mitochondria may be aging in similar ways across tissues and with similar temporal dynamics. However, by analyzing animals, tissues, cells, and mitochondria on an individual level, we observed many instances in which mitochondria changed asynchronously during aging, Fig 3DH Fig S3KM. For example, we observed that at days 7 and 9, some animals exhibited mitochondrial BA NADPH lifetimes in the hypodermis that fall within the day 1 lifetime distribution, while BA NADPH lifetimes in the BWM of the same animal fall within the lifetime distribution observed in day 11 animals, Fig 3G. Further analyzing anterior or posterior cells in the same tissue revealed that mitochondria in posterior cells of many C. elegans age faster as assayed by BA NADPH FLIM than mitochondria in anterior cells, Fig 3H. Thus, these results uncover a ubiquitous shift in mitochondria during aging beginning in midlife, as well as modes by which aging progresses asynchronously across biological levels. Finally, we asked whether the age-dependent changes we observed in mitochondria would be altered in response to modulating longevity. To this end, we imaged and analyzed WT and long-lived E2 at 1116, mutant C. elegans, a genetic model of dietary restriction, throughout aging analogously to our experiments in WT animals, Fig. 4a-b S4a-b. As we had found earlier that WT animals exhibited a strong shift in mitochondrial NADPH lifetimes during aging, we hypothesized that long-lived EAT2 animals would either display an attenuation of these changes or a different trajectory during aging. We found that up until day 11 of adulthood, EAT2 animals were largely refractory to age-dependent changes in BA NADPH FLIM endpoints across tissues, Fig. 4CF S4CE. For example, in WT animals, BA NADPH lifetime increases during aging, starting at days 5 to 7. However, in EAT2 animals, we did not observe a similar increase in BA NADPH lifetime. Although we observed a small increase in BA NADPH lifetime with age in some tissues, e.g. PM mitochondria, these effects were largely diminished when compared to the effect of age observed in WT animals. Further, we found that this attenuation of age-dependent changes to mitochondrial BA NADPH FLIM endpoints was matched with an attenuation of age-dependent changes to C. elegans mobility, Fig. 4g. Consistent with our data demonstrating an attenuation of age-dependent changes to BA NADPH FLIM endpoints, we also observed an attenuation in the increase in predicted age observed in WT animals in our regression models, Figs. 1-3, demonstrating that longevity is associated with a reduction in predicted age measured by BA NADPH FLIM of mitochondria, Fig. 4h. Finally, to evaluate the extent to which mito-NADPH age clocks could resolve potentiation in longevity across a diverse range of interventions which influence longevity, we examined BWM cells in WT, NHR49, NR2041, short-lived, CLK1QM30, long-lived and age 1 HX546, long-lived mutant C. elegans, Fig4IK, S4F. Consistently, we observed that NHR49 mutants which have shortened lifespan were associated with higher mito-NADPH ages earlier in life relative to WT, whereas CLK1 and AGE1 mutants which have extended lifespan were associated with lower mito-NADPH ages. Thus, these data demonstrate the sensitivity of mito-NADPH age clocks to changes in longevity. Discussion. Here we developed and applied BA NADPH FLIM in adult C. elegans to non-destructively track mitochondrial NADPH in individual mitochondria throughout cells and tissues in C. elegans across aging, and use the resulting data sets to develop a next generation of non-destructive, label-free, and cellular resolution aging clocks. BA NADPH enables both the ability to track the abundance of NADPH by tracking the fluorescence intensity of BA NADPH, as well as additional properties resolved by tracking the fluorescence lifetime of BA NADPH. As such, BA NADPH FLIM expands upon previously developed biosensors for imaging NADPH, which can only image the abundance of NADPH40 in living cells that our data suggest would not be predictive of biological aging. This approach to quantify aging is powerful, as its strengths complement the weaknesses of many previously existing orthogonal approaches 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 41, 42, 43, 44. For example, BA NADPH FLIM is fast, providing the capacity for a prediction of age on the same day as data acquisition. Further, BA NADPH FLIM can track individual mitochondria, providing the capacity to trace mitochondria during aging at cellular or subcellular resolution within an intact organism. BA NADPH FLIM is also label-free and non-destructive, thus remedying concerns that reagents used in the assay may be confounding results, or that the same individual could be measured at multiple time points throughout life longitudinally. As our approach relies on resolution of biophysical changes to NADPH in mitochondria, mito-NADPH age clocks also provide quantification of aging through a unique lens which diversifies our cohort of approaches to quantify aging. Thus, BA NADPH FLIM represents an approach to quantify aging with many powerful advantages over comparable previously existing technologies. While BA NADPH FLIM has many advantages, BA NADPH FLIM also has limitations. For example, as other species of autofluorescence eventually overcome mitochondrial signals in the BA NADPH channel in animals close to death, mito-NADPH age clocks have an upper limit in ages that they can track. However, future optical aging clocks could integrate these different species of autofluorescence to overcome this limitation. As NADPH lifetimes are a comprehensive representation of the biophysical environment of NADPH, and can be sensitive to changes in many physical and chemical parameters, such as enzyme binding, temperature, pH, and viscosity, it is also difficult to know exactly what the changes to NADPH we detect during aging in mitochondria specifically represent. Our initial investigation is also limited to analysis of C. elegans, as NADPH FLIM is widely used across many model systems 1645-46, and mammalian aging is known to be associated with changes to autofluorescence across many tissues. 47, 48, 49. Application of NADPH film to study mammalian aging is a promising future area of investigation in many model systems. Despite these limitations, the advantages of NADPH film represent a substantial advance in our capacity to quantify aging and provide a unique cohort of advantages that broaden the toolkit of aging clocks available to the neuroscience community. Taken together, here we describe a live cell, label-free, non-destructive, and subcellular resolution approach to track mitochondrial NADPH throughout a multicellular organism on the level of individual mitochondria. Using this technique, we spatiotemporally resolve components within mitochondria which change during aging across cell types and tissues, and develop models for the prediction of age, health, and remaining lifespan that expand our technical capacity to quantify aging.